Okay, good morning, everyone. My name is Alberto Ortin. In the first place, I'd like to thank the Associação Brasileira de Polímeros for organizing this webinar today, and also thanks to all the attendants. I will talk about separation techniques for polyolefin characterization, showing some recent developments, and also with an emphasis on new tools for process control and quality control. Let me start with some words to introduce my company, Polymer Char. We're based in the city of Valencia, Spain. And here in Brazil, we have already a long-time partnership with dpUNION, supporting our users both in sales, but also in technical support. They have been trained in our facilities, and we have very good cooperation with dpUNION. We have been dedicated to polyolefin characterization for almost 30 years. Our main business unit is development and manufacturing of instruments for polyolefin characterization, but we also provide analytical services from our laboratory, as well as training and consulting services. As being specialized in polyolefins, our customers are the polymer producers and processors mainly, but also those doing research in polyolefin characterization. Along the years, we have developed different analytical and preparative instruments for detailed characterization of polyolefins. But in recent years, we have also put our effort in developing tools for process and product control, which can be implemented in the manufacturing plants. This is the outline of what we will cover today. After a brief introduction, I will present some innovations that we apply to separation techniques. I'll introduce then the techniques for chemical composition distribution determination, as well as preparative fractionation. And finally, I will discuss on those new tools for enhanced process control, quality control. Very important. Polyolefins are basically produced using ethylene and propylene monomers. They are produced as homopolymers, polypropylene, polyethylene, but also as copolymers when an alpha olefin is added as comonomer, 1-butene, 1-hexene, 1-octene, or ethylene in the case of polypropylene. Even if chemically simple, made of carbon and hydrogen atoms, polyolefins are found in a large variety of applications due to their versatility and different properties which can be achieved by design with these materials. That versatility is due to the polyolefin different microstructural distributions, which can be controlled, such as molar mass distribution, chemical composition distribution, also stereochemistry, monomer sequence length, launching branching. Most important are the molar mass distribution and chemical composition distribution. The average properties measured by spectroscopic techniques are not enough to define a given product. So separation techniques, which can fractionate the polymer according to some microstructural feature, are required. Very important to keep in mind then is that polyolefins are polydispersed products, meaning they exhibit a distribution in molar mass, which can be broad or narrow, which in some cases is enough to characterize them, as in the case of homopolymers or homogeneous copolymers. But in the case of heterogeneous copolymers, there is also a distribution of chemical composition, which may be very complex, as in the case of Ziegler-Natta LLDP, we see in this figure, or heterophasic impact polypropylene and some others. So distributions are key. Polyolefin microstructure can therefore be characterized basically by both a chemical composition distribution here or a molar mass distribution. For some polyolefin cases, high-density polyethylene, low-density polyethylene, homopolymers in general, the molar mass distribution is more significant, while the chemical composition distribution is either narrower or has no special features. While in other cases, it is the CCD, the chemical composition distribution, which is more significant and so more useful to identify products and to relate to microstructure, or to relate microstructure to the product properties. In order to analyze those distributions, we need separation techniques. From the analytical perspective, in the dimension of chemical composition, most important are the crystallization techniques, Christoph, Treff, CF, while in the dimension of molar mass, we find GPC or size exclusion chromatography, which is the workhorse. When adding complementary detectors, GPC with infrared or TREF with light scattering viscometer, useful information in the interrelation of the two dimensions, chemical composition, molar mass is gained. Some other specific techniques, such as triple detector GPC, A4, intensive viscosity analysis, or also the solubles analysis by gravimetric method or automated Q-Stex, have been developed for special applications. From a preparative side, we have also methods for chemical composition fractionation or for molar mass fractionation, which can be complemented by analytical techniques, separation techniques, to study the bivariate distribution molar mass chemical composition, which is in fact the fingerprint of the polyolefin. That bivariate distribution can also be analyzed by two-dimensional analytical techniques, coupled techniques, CFC cross-fractionation, or which is a combination of TREF with GPC, or interaction chromatography with GPC. Now let's review some innovations in solution separation techniques related to sample care and to detection by infrared. All the separation techniques, we need to work with dilute solutions. In the case of polyolefin, that requires the use of specific solvents at elevated temperatures, over 140. This may be challenging due to safety and health concerns in handling the solvents and hot solutions. In addition, there are also analytical concerns due to the possible partial precipitation of samples, cold spots, lack of reproducibility, and so on. So automation of all the analytical process is highly desired, including the sample preparation. The analyst should only put the polymer dry in a vial and then bring those vials to the instrument. Then the solvent addition, dissolution, filtration, and separation detection at high temperature is done by the instrument with full safety. Automation of all those steps make a big difference in efficiency and also in reducing the analyst risks. In sample preparation dissolution, attention must be paid to thermal degradation. That thermal degradation reduces the observed molar mass and shifts the molar mass distribution, as we can see in the graph. We know that having an inert nitrogen atmosphere in the dissolution vial, this problem is delayed. It is also important to control the time spent at high temperature, reducing the mechanical stress, applying gentle shaking instead of stir bars, and also removing the oxygen during dissolution by means of a nitrogen part, as I said. We apply all those features in the instruments. Another big development in separation techniques for polyolefins has been the application of infrared as detector, especially in the case of GPC analysis of polyolefins, instead of the refractive index detector. We proved that the IR detector shows a linear response to polyolefins to same sensitivity to polyethylene or polypropylene or copolymers, as we see in this graph. Some words on how that works. This is basic diagram of a filter-based detector used in GPC and in other separation techniques. We have an infrared source, which generates infrared light with broad emission, a flow-through cell which is heated, and an IR sensor. IR bands are selected by means of optical filters in a wheel. There is a filter which selects IR radiation in wavelengths with absorption from the CH bonds. And the measured absorbance is linear with concentration according to Beer's law, as we have seen in the first graph. In addition, we have specific filters in the same wheel for methyl groups, which are found at the end of the searching branches due to comonomer, and for the methylene groups found in the backbone of the polymer. From those two filters, absorbance signals are obtained, and the band ratio is proportional to the methyl group frequency, which is related to the chemical composition of the copolymer. IR is a quite robust detection method, giving an excellent baseline, as we can see in this graph. This leads to enhanced precision in the molar mass distribution and also in the moments. Here we see 10 replicate analysis of GPC material by GPC with infrared. The relative standard deviation for number and weight average are around 0.6, 0.7, lower than 1%, which is quite good for high temperature GPC of polyethylene. And that was 3 to 4 times better than the results obtained by DRI detection in the same instrument. One quite challenging application is that of ultra-high molecular weight polyethylene. The high sensitivity and stability of infrared detection allows a drastic reduction of the injected mass, which is required when dealing with such high viscosity solutions. Here in this example, we injected 10 to 20 times lower mass than in standard GPC analysis, and still the signals, the peaks we obtained were good enough. So the IR detector is used with advantage as concentration detector in GPC, but most important in the case of high temperature GPC for polyolefins is that multiple band IR detector is also used as a chemical composition detector. In addition to the total polymer concentration, the IR measures simultaneously the absorbance chromatograms for methyl and methylene groups, as we have seen. The rates of those two signals can be calibrated to methyl group frequency, methyls per 1,000 carbons, and used to calculate the short chain branching and the comonomer fraction at every slice. So after GPC molar mass separation in the GPC columns, after that molar mass separation, the IR detector identifies the chemical composition, which is in different dimensions as the separation. In this way, GPC with IR allows a study of the chemical composition along the molar mass distribution, giving a more complete picture of the polymer. With the current trend in industry towards more complex heterogeneous products, the information on chemical composition against molar mass is really of great value. Here we see two representative examples. In the left-hand side, we see the analysis of two similar bimodal high-density polyethylene products with a small amount of short chain branching added in the high molar mass fraction. Short chain branching we can read in this graph. One of them was 3 short chain branches, the other around 6 short chain branches per 1,000 carbons. Those levels of those small amounts of short chain branching can be quantified with infrared, and the different levels can be related to the final product mechanical properties, particularly the environmental stress crack resistance. And also another interesting application here we see in the right-hand side to ethylene propylene copolymers, which have similar molar mass distributions, but the ethylene fraction distribution along the molar mass is quite different. One of them, the one in green color, is quite uniform. This is the ethylene content around 10%, as measured by the infrared, while the other in red color, we see that it's more heterogeneous and has a larger content of ethylene in the high molar mass fraction. The IR detector is the only detector that can give this information coupled with GPC. Also, the short chain branching trend given by the IR helps in identifying the catalyst system or study the catalyst. Here we see GPC IR analysis of Ziegler-Natta LDPE showing a clear drift in short chain branching on the molar mass distribution. There is a lower number of short chain branches in high molar mass, and there is a trend increasing short chain branching to lower molar masses. This is in contrast to the uniform comonomer incorporation of single-site catalyst products. So we see three different single-site catalyst polyethylene, different levels, but they are rather uniform. Okay, so let's change topic now to crystallization techniques. Those are used specifically to study the chemical composition distribution CCD. This CCD, in many cases, is more discriminating feature than the molar mass distribution. chemical composition distribution is measured in a temperature scale by crystallization techniques. And in the case of polyethylene, that is, temperatures are related to the number of sorting branches. Sorting branches reduce the crystallinity and so reduce the crystallization temperature. Here, in this example, we see the case of a multiple reactor LLDP, in which there are several degrees of freedom by product design by playing with the catalyst or process conditions, varying the amount, the relative fraction of each component, or also varying the amount of comonomer added. So the temperature would shift or the shape of the distribution will change. However, even if all those features are reflected in the chemical composition distribution very clearly, in the molar mass distribution, they have a very small impact. So in this case, separation by chemical composition is required even more than by molar mass. For polypropylene and ethylene propylene copolymers, the CCD is related to irregularities in the chains, which also reduce the crystallinity and reduce the temperature. For example, incorporation of ethylene as comonomer, but also the tacticity or regio errors. Here we see a typical distribution of a heterogeneous impact copolymer with a peak at high temperature due to the most crystalline isotactic polypropylene. Then some crystalline propylene-rich EP copolymer, even some pure polyethylene peak we can obtain sometimes, and then an amorphous fraction, higher level of ethylene, EP rubber, observed at room temperature. So differences among products can be clearly discriminated by the chemical composition distribution, while the molar mass distribution has little information. There are very small variations in the molar mass distribution. Okay, so Christoph and Treff are crystallization techniques, both working in dilute polymer solutions. As we can see in this figure, for this analyzing of the same material, polyethylene in this case, by Christoph and Treff, both techniques provide equivalent results for the chemical composition distribution. Christoph is running a vessel with no support and is measuring the cooling cycle during the crystallization of the polymer, while Treff is running a column and data is obtained during the heating, the redissolution. So due to the polymer undercooling effect, this causes differences in the observed temperatures. So there is a shift to higher temperatures in Treff, as we can see here. That shift is larger for polypropylene than for polyethylene. For that reason, some polymer combinations are not resolved by Christoph, as we can see here, but they are separated by Treff. That's the case of isotactic polypropylene and homopolymer polyethylene. That can be resolved by Treff, and Christoph depicts would coelute. But there are other combinations of ethylene propylene copolymer plus homopolymer polyethylene, which are properly separated by Christoph, but in Treff there is no separation. So depending on the combination, Christoph or Treff may be superior, while in general they are equivalent. There's a more recent technique is CEF. CEF stands for crystallization elution fractionation, and in CEF, a dynamic crystallization, which is in a way equivalent to Christoph, is combined with a Treff separation. This combination results in improved separation, taking advantage of the separation power of both modes, Christoph and Treff, in a single technique. As an application, we see here the analysis of two polyethylene products which are uniform but have slightly different densities. There's better resolution with CEF than with regular Treff when used in the same conditions, in the same analysis time. Those two polyethylene copolymers with similar densities, in CEF the separation was 20.7 degrees, which is larger than the separation obtained by Treff, only 12.7 degrees. So separation power of CEF is larger than Treff. But CEF can also be used as a high throughput technique performing much faster analysis and still keeping reasonable resolution and precision. Here we see the 10 overlaid analysis of multiple reactor polyethylene resin with only 25 minutes analysis time. So we can see clearly the resolution is good, precision is great, and the analysis time is very short. So the CF can either be used as a higher resolution or fastest technique compared to TREF. Now, let's turn our attention now to preparative fractionation. Preparative fractionation is a very powerful tool for research and development. It is true that it requires much more resources and analytical tools, and also a high level of expertise. So in many scenarios, outsourcing is still a best option. We have developed two different automated instruments for different scales, and also have been working to develop the expertise in several fractionation modes for different applications. So we have there is a system for small scale fractionation, another for larger scale, and different fractionation modes by TREF, by Christoph, even TGIC, by molar mass. We will see this now. The first preparative system, the fractionation takes place in a stirred vessel with no support. In fact, in the instrument, there are two vessels, so two samples can be fractionated simultaneously. The amount should be lower than 2 grams per vessel. It can work both in TREF and Christoph modes for chemical composition, but also by mixtures of solvent and non-solvent for molar mass fractionation. In the TREF mode is performed at increasing temperature steps, and it works well to obtain fractions of different chemical composition. We see here in black color the TREF analysis of the parent sample, a broad distribution sample, polyethylene sample, overlaid with the analysis of the different fractions taken at different temperatures. They are narrower distribution fractions. For a larger scale fractionation, we develop a column fractionation apparatus, which also automates the fractionation process. And depending on the kind of column installed, it is used for TREF fractionation with inner packing, molar mass fractionation, even interaction chromatography modes for lower crystallinity polymers are possible. The solvent consumption is rather large, 5 to 10 liters per fractionation, and also the time for full fractionation could be 1 or 2 days. But it can handle up to 20 grams of polymer. Here we see a diagram of the instrument with a stainless steel vessel for dissolution, and then a large column for performing the fractionation, a series of collection bottles outside of the instrument to collect the fractions. There is a pump, which could be a regular isocratic pump for TREF, Christoph mode, but also a quaternary pump to prepare solvent mixtures when required for molar mass distribution, molar mass fractionation. An example of TREF fractionation starting with 18 grams, in this case, of an LLDP, and collecting 11 fractions is shown here in this figure. Those are the analytical TREF of the fractions. As you can see, they are quite narrow fractions in chemical composition. Also, molar mass fractionation on a column with thinner support with solvent and non-solvent mixtures is possible. Again, here we see the molar mass distribution of the parent sample, which is broad, and then the molar mass distributions of the narrow fractions overlaid. So now, in this second part of the webinar, I'd like to introduce some new analytical tools we developed for process control and quality control in the manufacturing plant laboratories. Polyolefin production, we know it takes place in large capacity facilities with where there are different types of reactors, where we find different analyzers for monomers and others. Then in the plant QC laboratory, the product itself is controlled by measuring some properties such as melt index, density, additives, and also the solubles in the case of polypropylene, and that information can be fed back and used for process control. We know that those properties, melt index, density, are in fact related to microstructure. Microstructure means molar mass distribution as well as chemical composition distribution, as we have seen. However, that detailed microstructural characterization is done in analytical and research laboratories, which typically are far from the manufacturing plant. But distributions are key, as we have learned. So the challenge is, can we obtain the distributions information in the plant laboratory to better support manufacturing? So those distributions, is possible to obtain them in the manufacturing plant laboratory, and that would allow more precise control, going from melt index to molar mass distribution, and from density to chemical composition distribution. And also the determination of the solubles can be automated, and also including online multiple detection for ethylene and intrinsic viscosity, making it a very powerful method. There is a need for that more precise control. I will explain that. In this figure, we can see different polyethylene materials that, in spite of having same melt index, similar melt index, and also very similar densities, they have completely different molar mass distributions, as we can see in the figure. And so completely different performance and processing behavior. Another good example is that of pipe resins, bimodal resins, or in general multimodal resins with complex structure and multiple variables to control. It's not possible to really define the product and control the process only relying on the melt index, which is related to the average property, not the distribution. So with today's sophisticated industrial resins with optimized performance in specific applications, melt index and density are very far from defining the resin performance. So there is a need for enhanced process control based on the full distribution. Taking that challenge, we work to develop some instrumentation for process control, QC, in the plant laboratories. Design principles are different than we found for analytical instruments for research. So here the design principle were simplicity and ease of use and automation, reliability and safety, minimum solvent consumption, fastest analytical approach. That's important to be able to move the information to the process control to keep it working in control, and reducing the production of a gray material, and with minimum lab bench space as well. So with those principles in mind, we developed GPC QC as a simplified GPC instrument optimized for the manufacturing laboratory. Given the application for GPC QC is following the process changes in manufacturing, it demands the analysis of a single sample of material which is produced at a particular time. Then sometime later, another sample from the process is coming, and so on. That means that there is no need for a sophisticated autosampler with many vials, but a single sample dissolution station, which is optimized to shorten the dissolution time by filling automatically with preheated solvent and applying vigorous stirring. Sometimes, depending on the process, a significant amount of sample may be required to get good representativeness. Large capacity vials of 100 or even 200 milliliters are used in this approach. Up to 200 milligrams of polymer can be dissolved for analysis. Shortest analysis time is possible by using single fast column, GPC columns. Both time and solvent used are minimized. Reliability is improved by simplifying the hardware and using robust detectors. The analytical workflow is quite simple and requires minimum manual work. When a sample of polymer is received in the laboratory, it is weighed and into a disposable bottle, which is placed into the dissolution station oven. Then the analysis started from the computer. The needle is inserted through the septum, and it proceeds automatically through filling, dissolution, and injection to the column according to a preset method conditions. When a new sample comes in, a new bottle is prepared with it, and the analyst just discards the previous one, placing the new one in the station. Following this workflow, the GPC-QC can be operated continuously with a cycle time of 1 hour or less, which is found appropriate for controlling the startup of reactors or when change of grade is conducted. Once the process is stable, production may need to be controlled at a slower pace, maybe 1 or several times per day. It's very precise. We see here in this graph 7 overlaid chromatograms. And in the tables, we see how the precision for MW and MN are really outstanding, and even for polydispersity or all the relative standard deviation lower than 0.7%. So this tool may complement the existing control based on melt index and density, but with more complex resins, having the full molar mass distribution is becoming actually a need. As an application, we see here 2 copolymers with their molar mass distributions, and also the sorting branching thanks to the IR detector. So from the molar mass and the IR detected sorting branching, it is possible to estimate melt index and density as well, if that is necessary. As I said before, a very interesting application for that technology is that of production of bimodal high-density polyethylene made in dual reactor process. Lower molar mass high density is typically produced, and a larger molar mass with a small amount of sorting branching is added as with some comonomer, and that balance will result in enhanced mechanical properties for pipe applications. From a single GPC analysis, and in less than 1 hour, with minimal operator intervention, it is possible to obtain the molar mass distribution, so in the relative weight fraction of the 2 reactors, also the polydispersity, the molecular weight of each of the which is produced in each of the reactors, but also the level of comonomer and how it is incorporated along the molar mass distribution. In this case, the sorting branching at the high molar mass fraction was quantified. So we can see here that several variables can be used for control of the production of this type of products. As I said, the weight fraction of each component may be changed, their molar mass, polydispersity, branching distribution, or comonomer content. Most of that information is not available when measuring only melt index and density. So we're sure that companies will benefit from implementing this technology in manufacturing. Some companies already are doing, in order to reduce the upgrade production and also contribute to a smoother startup and transitions in reactors. Yet another application for GPC-QC may be found in the production of complex ethylene-propylene copolymers, measuring the molar mass distribution, but also the ethylene content is important to control. However, for the production of polypropylene, the so-called saline solubles is a fundamental parameter. It provides a measurement of the small amounts of undesirable low tacticity or low molar mass fraction in homopolymer and random copolymer resins, which has a significant influence in polymer performance. In the case of high-impact polypropylene, an amorphous rubber phase is produced on purpose to give impact strength, and that the control of that fraction is essential also in the production of those resins. The molar mass through the intrinsic viscosity of the rubber and total material are also important for some products, as well as the ethylene content in the rubber phase or in the crystalline fractions in copolymers. In the case of homopolymer PP or random copolymer, the solubles consist of the atactic polypropylene, as I said, or amorphous ethylene propylene copolymer. In some other processes, dual reactor for high-impact polypropylene, that rubber phase is produced in a second reactor, and the solubles are mainly related to that PP rubber phase. In those products, we find a crystalline matrix made of, in one of the reactors, of isotactic polypropylene, and then a dispersed amorphous phase, which is measured as the solubles. Historically, in gravimetric method, ISO or HTM have been used. Despite requiring significant manpower, handling of solvent, long analysis time, especially the viscosity or the ethylene content need to be measured. So there are many manual steps involved, and that may affect adversely the precision of the method. So Crystals QC is an automated method based on TREF, which is used to separate the amorphous from the crystalline fractions. The polymer solution is loaded into a TREF column. Then the temperature is decreased very fast to near room temperature, so the polymer crystallizes after we allow some time there, and a pump is started to elute the soluble fraction as a peak. The soluble fraction is moved towards the detectors. Once the soluble fraction is taken out of the column, the temperature is increased again, and with the flow stopped to redissolve the crystalline fraction within the column. After some time, it is eluted again, turning on the pump towards the detectors. After the column, which sits in a micro oven, there is an IR detector and online viscometer. So in this automated analysis, the amount of soluble is determined, but also the ethylene content and the intrinsic viscosity of the solubles and the crystalline fractions, as well as the bulk material. So we can see here for different results obtained for different samples. For the soluble fraction, the amount of solubles, the ethylene content, the intrinsic viscosity, and also the crystalline fraction. High precision is obtained due to the full automation of the method. Here we see the measured solubles for two samples of very different levels, around 4.5% and only 0.1% variation. And second copolymer with larger solubles of around 28%, still with very low error or variation. Also, in the case of homopolymers tend to have very small solubles fraction. The precision is quite good. In this case, we have a homopolymer with less than 2%, and the precision is very good. As I said, in addition to quantifying the soluble fraction, the intrinsic viscosity for the whole polymer, the soluble, and the crystalline fractions are measured. For this sample, we have here replicate analysis for the same sample. And we can see how the intrinsic viscosity for the soluble fraction is 2.4, which is larger than the intrinsic viscosity of the crystalline fraction or even the whole. So that may be important in some applications. And the IR detector gives the ethylene content, which is around 32% for the soluble fraction, the ethylene propylene rubber, while it is quite small for the crystalline. There's still some crystalline ethylene propylene copolymer, but very low content. Before I finish, I would also like to take a small time to mention a new fully automated intrinsic viscosity analyzer. This system can be used for solution viscosity analysis of any polymer soluble from room temperature to 200 degrees centigrade, especially useful for ultra-high molecular weight polyolefins, which are very difficult to handle with other semi-automated or manual methods. Also for other polyolefins, polypropylene, polyethylene in general, but also for PET, PLA, and others. And it's compatible with many different solvents, TCB, ODCB, but also decalene, tetraline, acetone, even water. It is based on a very robust two-capillary relative viscometer detector. And for polyolefin applications, the infrared detector is coupled and is useful to quantify the amount of polymer injected, so there is no need for accurate weighting. In different applications, we found good correlation to existing alternative methods for polypropylene or polyethylene comparing to the ISO 1628 method. And also recently we ran some analysis of cellulose ester in acetone at 30 degrees compared to other methods, and the results are well in line. So that brings me to the summary of the presentation. We have seen the importance of sample preparation because we need to dissolve at elevated temperatures, so we need to take some precautions, reduce the dissolution time, control the dissolution time, add nitrogen to purge and remove the oxygen during dissolution. That helps to preserve the high molar mass fractions. Then the GPC with higher detection, which is becoming a standard technique given the potential of multi-reactor resins, copolymers, polyethylene, polypropylene combinations. We have seen that the chemical composition distribution is most discriminating microstructural feature in many polyolefin types. Preparative fractionation, which is a very powerful tool for R&D, also, although it's a resource-intensive technique. And finally, I also emphasize that there is a need in the industry for enhanced process control based on the full molar mass distribution, MMD, instead of relying on average value melt index. And for that application, we developed the simplified QC-dedicated GPC, which is available. For polypropylene and ethylene propylene copolymers, a precise and automated method for solubles determination is also required. Multiple detection expands possibility for control, adding the ethylene content and the intrinsic viscosity of the fractions and the total. Okay, and with that, I finished the presentation, and I think we have some time to go through some of the questions. Here you can see the contact information for dpUNION, our partner in Brazil, as well as mine, in case you want to contact later on with me directly.